So good afternoon. It's indeed a pleasure to be here to introduce our speaker, which I would introduce formally in a minute. But just to start, two housekeeping things to remember. The CME code is 57660. And for those watching online who would like to submit a question, please hit the Send Live Feedback button, and your questions will be relayed to the speaker at the end of the lecture. But you can submit questions at any point. Now, before I introduce our speaker, I want to really honor that he's actually here with his wife, and I would like to say a few things about the wife, who is a researcher and is a very successful professional on her own, Professor Anissa N. Happy. She holds a doctor of veterinary medicine degree, a master's degree in veterinary science, and a PhD in veterinary pathology from the University of Ibadan in Nigeria. She's also a fellow of the College of Veterinary Surgeons of Nigeria. After her doctorate degree in veterinary medicine, and Dr. Anis Happy obtained a World Health Organization Fellowship for postdoctoral training in molecular biology at Harvard University in 2002. She joined the Redeemer University in January of 2021, and as the head of zoonotic research and surveillance, she has been instrumental in the success of this center. And I just wanted to say, please, can you stand up, and thank you for coming. So I'm going to quickly do a short introduction of Christian Happy, our speaker today. That's about the best I can do given the time. I don't want to take all of his time, but you can see his work if you Google him or go on PubMed. He's a fantastic scientist. But at a very personal level, when I met Christian some years back, I was so impressed, and I told myself, like I told my lab this morning, that this is somebody to watch out for, and that he's going to make a difference. He has exceeded that expectation in a very dramatic way. Francis and myself visited him recently, Francis Collins, to open a new center of genomic science. And that center immediately was elevated to an institute level. And I'm going to talk a little bit about it. I'm sure you will hear quite a bit of it in Christian's presentation today. So Christian is a distinguished professor of molecular biology and genomics, and the founder and director of the African Center of Excellence for Genomics of Infectious Diseases. And that's the one that I say has just been elevated to an institute level at the Redeemers University in Ede in Nigeria. If you see the location where this center is, that's when you actually fully appreciate what he has done, in a sense, to bring that kind of center to that kind of environment is truly, truly beyond words, and I'm absolutely glad. It's also an extension of a vision in terms of H3 Africa, a program that he fully participated in. He did his postdoctoral fellowship at Harvard University, and he's currently an adjunct professor at Harvard T.H. Chan School of Public Health. His research is on human and infectious disease genomics, with focus on host pathogen interactions. Professor Happy pioneered real-time genomics epidemiology, pathogen genomics evolution, and open data sharing to guide public health response to outbreak in Africa, starting with the 2014 West African Ebola outbreak. He and his colleagues use a translational genomic approach from Ebola virus genomic sequences to quickly develop first Ebola rapid diagnostic test in the middle of the raging epidemic of Ebola then in West Africa. The real-time outbreak response system that he and his colleague established in 2014 was effectively used by the global public health community to respond to the COVID-19 pandemic. Professor Happy and his team, in an unprecedented way, used next-generation sequencing technology to perform the first sequence of SARS-CoV-2 in Africa, within 48 hours of receiving the sample of the index case in Nigeria. This seminal work inspired and built confidence among African scientists to detect new variants of SARS-CoV-2 and share the virus sequence data with the rest of the world. This paved the way to the development of new countermeasures, including CRISPR-Cas13 diagnostic development, in his lab. He very recently used a GWAS to identify novel human genetic variants as associated with resistance to Lassa fever infection, as well as the fatal outcome to infection with the virus in West African populations. Again, working with his colleague, they have successfully now leveraged CRISPR-Cas gene editing technology to develop a rapid diagnostic test for Lassa fever and for Ebola virus. Through the Genomic Institute that he has, he established, he has had a huge impact across Africa in terms of genomics and infectious diseases. He single-handedly, with his colleagues in the center, have trained over 2,000 scientists from 53 of the 54 countries across Africa. That is truly, truly, truly remarkable. Professor Christian Happy is recipient of numerous awards. I'm not going to go into detail, but I just want to highlight that just a couple of days ago, he became a member of the U.S. National Academy of Medicine, a fellow of the Nigerian Academy of Sciences, and a fellow of the Academy of Medical Sciences of Nigeria. Christian, please come.
Well, thank you, Charles, and I want to get started by thanking the director of NIH and then the director of NIGRI and Dr. Fogarty for your presence. And I want to thank everyone for the opportunity for being here and to give this speech. So I think it is an honor and a privilege to be here because I know this process is highly vetted. And today I think I'm just going to be sharing with you a little bit about how we've been able to pioneer genomics or use genomics as a way to perform surveillance and then to characterize microbial threats in West Africa, and how effectively we've been able to use that to actually contain some of those disease threats, not only in Africa, but in West Africa. So, and full disclosure, so I lead the center that has become an institute right now, and then we are as a reference center for WHO and Africa CDC for infectious disease research and innovation, but we're also a center of excellence for the World Bank, and as such, so just to the least to say. So as we all know, we're all talking about genomics, but we need to know that that's really technology or that approach to medical research has really paved the way or transformed the way we approach healthcare, and then research. But then we also realize that, in as much as, that lead to major breakthroughs and then a lot of innovation. So we realized a few years ago that that also created a huge divide between, the rest of the world and then Africa. And then the reasons for those divide was very clear. Lack of human resources, lack of infrastructure. And then as such, it was very evident for some of us that if care was not taken, Africa was going to miss this train of this novel revolution for science. And what we did was basically to start thinking about how do we, what can we do to avoid such an opportunity in Africa. And we looked at the landscape, and what we realized is that, Africa is not only blessed with natural resources, but is also blessed with human resources, and then also very powerful biodiversity. And as part of the very powerful biodiversity in Africa, so you also see what I call the pathogens. If you look at this map, for instance, you realize that, about 70%, just as in passing, of pathogens that are responsible for pandemic or potential or epidemics are all located in Africa. So that for us is actually wealth that we could actually exploit in order to address not only African challenges, but actually global health challenges and address the health security of everyone. So in order to actually address this major problem that could just not only make Africa participating in the global revolution, a genomic revolution, but also to give the continent a voice, we basically decided to focus on infectious diseases. But how could we have done that? We decided to set up a platform, called the African Center of Excellence for Genomic of Infectious Disease, from a World Bank call, and then based on long-term, research and training partnerships. That is leveraging the relationship that we had with academic and clinical testing centers in West Africa with partners across, the Atlantic Ocean. So naturally Harvard and then Broad, and then also with the private sector. So, and then we had actually set up a mission in order to bridge the gap that I mentioned earlier on. So we really realized that there were two things that we have to do. One was actually to educate and then build, the critical mass that we needed in order to give people the impetus and then the knowledge that they did. So creating an academic and vibrant research environment that will basically just, where we'll do relevant, and high and translational genomic research, in Africa. But in order to do that, we needed to provide people with the right skills and knowledge. So, and then therefore we created genomic programs, the very first genomic programs on the continent that had the responsibility to actually build a critical mass of well-trained African scientists that can harness and then leverage the skill and knowledge of genomics in order to address local problems, which is infectious disease, toward control and eradication. So we set up a master's and PhD program, internationally accredited, but we had to find a problem that cut across and that can bring everybody together. And then that problem that could bring all African scientists together or people across the continent together was the issue of infectious disease. And that really was for us to understand what is that thing, those problems that unite us in many ways. The whole idea of infectious disease became very important. And then we said, okay, in the context of Africa, most of the time when people have fevers, they go to the hospital, and then the first culprit in our context is often malaria. So when the doctor doesn't say malaria, then he says the next thing is typhoid. And in our context, because you see a doctor that is seeing 200 patients a day. So when you come around and then you have a fever, so he tells you that you have malaria typhoid. So which means in that case, he bombards you with both anti-malarials and antibiotics, so that at least you can go. But then the whole idea behind our program was to actually sit down and then identify the drivers of febrile illness and then use those information to create the next generation diagnostics, vaccine, and whatnot, therapeutics, and then make life of the clinicians easier. So in doing that, we're addressing one of the major critical needs for genomics in Africa, and then that is basically genomic capacity building. That is setting up the necessary infrastructure to do genomics, set up diagnostic cores, and then also create a sustainable environment for science and then for careers in this field. This for us was very important in order to reverse a trend that we've seen over the years, which is brain drain. But then is how do we set up a platform that can actually help reverse the brain drain and then actually make brain gain for the continent? So you will see how we successfully did that in the next few years by just taking one use case or two use cases. So, and in doing this, we're basically doing what we call, building the next generation of African pathogen hunters. That is, making these guys or the young ladies and girls and boys on the continent to hunt these pathogens in the dirt roads, in the bushes, characterize these pathogens, and then create the necessary countermeasures before they come to us. You're going to agree with me that as a global health community, what we're playing today is what we're playing defense. That's the truth. It's a fact. We just keep being reactive. We are not being proactive. And I believe that with the skills and the knowledge and the technologies available today, we can basically, be ahead of the curve and playing offense. That is basically leveraging that biodiversity, characterizing this pathogen, creating the necessary countermeasures before they get to us. And that was the whole idea and then the whole philosophy. We established this center in 2013. And then a few months after we established this center, we have what I call our first baptism of fire. So that is basically setting up a center with a very, very, very ambitious goal. And as you are setting up, then we had the very first Ebola outbreak. And then because we were funded by the World Bank, we were told, you say you want to do it and let's see it. And we really had to innovate and do a lot of things in the midst of an outbreak that nobody actually thought. And the very first thing we did when we had this Ebola outbreak was the fact that we really thought that, and we recognized one thing. And that simple thing was the fact that, the world is going to look away, but then what are we going to do to address the problem? And we realized that, if you do a SWOT analysis and you realize what is our weaknesses, the weakness is diagnostics. You cannot treat a disease or contain a disease if you don't have the ability to diagnose it. And in doing that, we went on to train the first set of people to actually do molecular diagnosis of Ebola. We trained in Sierra Leone, we trained in Nigeria, in Senegal, in other countries, and as we rightly, predicted, because we knew that this was going to spread over. The reality is that you can lock down the border, but you can't prevent people from moving. The borders in Africa are very porous. So what happened? People carry the viruses and the disease with them. So, and they don't need passport, they don't need visas, so they will have to move. The virus will move along with people. So, and it was already predicted, by May 20, 2014, we got a very first case, or suspected case of Ebola in Sierra Leone. And then one of the, Augustine Goba, one of the trainees of ours, actually did the first detection. But most importantly was the fact that we went from not just detecting, but to go ahead and then start doing contact tracing, eventually put the indexes toward the traditional healers, as you see here. And this guy who was thought that he was invincible was the root cause of spreading the virus. He was treating people. He died along the line, and then had actually created a lot of havoc. So within the first week, we actually suspected 35, confirmed 14. But what is important for us is the fact that we went ahead and then did something that was unprecedented, which is in the midst of an outbreak, we actually generated the first 99, viruses, or genome sequence of the virus, and actually shared it, to the global health community. So I think we were setting up a system that nobody knew or nobody has ever done. That is doing sequencing in real time, but also releasing the data in real time. And then pioneering what we call open data access, open data sharing during an outbreak. And that really becomes, the first leg or pillar of this model that Charles actually mentioned. But then apart from doing this, we basically went on to actually set up a protocol that we also shared globally, and then doing it very fast. Imagine 10 years ago, doing genome sequencing, whole genome sequencing in the midst of an outbreak. It used to take longer, but we actually improvised and then create a protocol that can actually help us to do sequencing much more faster. And then it took us 10 days from sample collection to sequencing and then depositing this in gene bank. And that our publications, again, came out like 9 months later. So this is basically we're showing the world that, in the midst of an outbreak, what we need to do is not just simply generate data, but also share the data for global public good. So that was the very first pillar. The second thing that we did is, in the midst of that outbreak, we made some very, very important observations. If you look at the map here, by June 16, we realized that SL3, this is a variant in Sierra Leone, has taken over. And then the question we're asking is, why is it that you have several variants circulating and then one actually masked the others completely? And then we went on to actually study it, far much better. We observed that there were just so many mutations, up to 371 mutations accumulated, 21 across, that were almost fixated. And those 21 mutations were associated with increased transmissibility. So we're discovering the second concept, that is increased transmissibility, variants of concern. So we actually pioneered that, but then what is important about, discovering that is the fact that we went on to actually, use genome sequence data actually to show how epidemics can actually spread from blocks to block, counties to counties, village to village, district to district, and eventually from within the country and then across the nation. This, again, was the first time where you could use genome sequence data to identify hotspot and then look at transmission chains, and then just only between country, but across country. And this data was shared in real time with national public health authorities, and then they used that as a way to actually start addressing the response. But as you're doing this, you're also facing some major challenges. One of those challenges is, how do you do it when you are actually having some PCR system and then it's taking forever to actually analyze? We realized that it was taking us 96 hours actually to test a sample because the backlog was just so much. So, and then we found that it was important to move from just PCR, but creating a method that can enable to diagnose much more faster and then get the result. Because along the line, we realized people that had malaria fever, because they had a fever, you would go to Ebola center and then get infected and take the disease back home. So in order to curb that, we needed to have like some kind of near real-time diagnostics. And then we set up the third concept, or the third pillar of, genomic, the third pillar for epidemic response and effectivity. So we brought in the whole concept of translational genomics, that is going from genome sequences to create the very first diagnostic that was approved by the US FDA under emergency use authorization and then the WHO. So that rapid diagnostic test that we never used to get the result within 10 minutes became what I call the turning point to contain that epidemic. But I can tell you that it wasn't easy, because at the time, you're dealing with organizations, I won't mention them here, that were used to do things in a way in the past 45 years or in the past 50 years of their existence. They never understood the value of doing things in real time. We were used to a system whereby when there's an epidemic, some expert will come in, take the sample somewhere, and after 2 years, they will tell you this is what happened in that epidemic. Here, we were providing the data in real time. And that made people very uncomfortable along the line. And they were like, no, it's not possible. You are doing science during an outbreak. This is not useful. These are all of the things that we were very dogged into the way we're doing it. And we provided this information. The good thing is that along the line, you got some leaders that could understand the value of what you're doing. And then I'll take the case of one country, where the head of state understood the value of what we're doing, and his Minister of Health was not aligned with what we're doing. And in order to solve the problem, the head of state fired his Minister of Health and said, well, I think what these guys are doing is correct. And effectively, it was proven right. But then what is important is actually to show how this works and then could do this in great time. It is a situation where this is Nigeria. This is our country, and this is a church. This church was about two minutes, practically from my lab at the time. And I took this picture personally. And then you go here, and on a Friday, you're getting 3 million people gathered in one single place in the midst of an outbreak. And you were wondering what would happen if you have just a few people in this hall, or in this gathering that have Ebola. And these were things that were running through our mind, and then at the time, of course, we advised that we didn't want to see these gatherings, but that gave us time to prepare. And we actually, in the case of Nigeria, got prepared, got the public authorities prepared, and in the case of Nigeria, we will see this system became very important. Because in the case of Nigeria, unlike what happened in Sierra Leone and what happened in Guinea, for instance, where samples were sent to Paris and then the result was sent back, a month later, and then transmission was spreading. In the case of Nigeria, within 12 hours or 6 hours, to be more precise, we had the result. And this result was shared in near real time. And we were able to track down all the patients or everybody that came into contact with that index patient. We were able to isolate them, monitor people. And I can tell you that, we did what was unprecedented. Working hand in hand, providing results, diagnostic results and genomic data in real time with public health authorities. Nigeria went in the annals of public health. Containing a disease like Ebola. We started in a city like Lagos, 25 million people. In a country of 230 million people, we were able to contain that in 93 days with only 20 cases and 8 deaths. That's really showing when a country is ready to absorb innovation and technology, they can actually beat all the odds. Because I've always said that you need 2 major things to beat an epidemic. One is speed, the second one is accuracy. Once you have those 2 things, speed of execution and then accuracy with your data and fact, you should be able to contain that, if you work hand in hand with authorities. So this is basically just giving you how you could look at contact tracing and then the genomic work that we're doing. But then that gives us a very nice story about what the story about Ebola was. But then I will go back and talk about one thing I was working on before the Ebola outbreak, because Ebola came and I considered it as a distraction at the time. This is Lassa fever. It's a virus that was described for the first time in 1969 in Nigeria, and that before I started working on it for 37 years, had no diagnosis on ground. But then we decided to go into it and then start working on that. And within 6 months of showing interest in this disease, we were able to do something that again was unprecedented. That is basically sequence the first set of virus. A whole genome sequencing of this virus, but enabled us to identify not only the ancient origins of this virus, but also help us to actually create rapid diagnostic tests. We found that this virus, even though described in the first time in 1969, was actually much more older than we ever thought. The virus emerged from present-day Nigeria about 1,187 years ago, was circulating in Nigeria for 600 years. moved out of Nigeria about 400 years ago to West Africa. And then that explained the reason why when you look at the case fatality rate and then the disease progression in Sierra Leone and compared to Nigeria is actually different because, the virus is much more younger in Sierra Leone and then there's no adaptation in that part of the world. But it went on not only to characterize that, but also to go on and then use that to create, the diagnostic that is needed. But again, to say that the diagnosis wasn't funny because you look at other countries in West Africa, you got only one particular lineage circulating. But in the context of Nigeria, you have 3 particular lineage circulating. I usually joke, by telling my colleagues in Nigeria that we say Nigeria is complicated. Not only human beings are complicated, even our viruses are complicated. Because you got to deal, and then it took us much more longer time to create a diagnostics in Nigeria because we had to think about creating a pan-virus, pan-Lassa diagnostic tool. But then moving forward, we also kind of leverage knowledge of, CRISPR-Cas, as Charles was mentioning in his introduction, but then using gene editing technology to make the diagnosis much more faster and then much more accurate and then a little bit more cheaper. And this was the very first CRISPR-Cas for an infectious disease that was done. So you see a report from Amy Maxson from Nature. She embedded in our lab for about 5 days and it really just to see how this was working. And then you could basically just so from the beginning to the end, and it is how our diagnosis occurred. But when you got such a diagnosis, the question is, well, yes, we were able to develop diagnosis, but then the question is, how do people respond to the disease? And that's where, we really started trying to ask the question, immunologically, what is the immune response? And then again, I mentioned brain drain or reverse brain drain at the beginning. So I think here I just want to give a shout out to one of my mentees, Dr. Chinedu Ugu. He's here in the audience. So I think I gave him this assignment. I think he's one of the active fellows now at NIH. And I think I gave him this assignment because he wanted to know a little bit more. He created one of those first, T-cell assays, actually, to understand exactly what is the immune response to those patients. And he did beautifully well by actually enabling us to really understand exactly what it is. Finding cross-reactivities for T-cells, across the different sites. But then that took a lot out of him because we also understood that, depending on where you are, you need to use different peptides. So, and that for us was very important and added a lot of value into the field. Now, so when we were dealing with this kind of thing, so you also realize that right in the field we're facing a challenge. We realize that in the field or in hospital, there are people that do have Lassa fever, and no matter what you do, their outcome is death. And then you also realize that some people do have the disease, but eventually they come out well. But then doing the work in the field, you realize some people do have the disease, tested positive for PCR, but they're ambulatory, and they have no symptom, and then they're asymptomatic. So the question was, what is really driving the fact that, some people have they are condemned to die when they have a disease, while other people are hanging around with the virus and then they're going around as if nothing's going on. So we went on to do the very first GWAS, for this particular group. It took us quite a number of years. But what is interesting is that at the end of the day, we came up with some very, very powerful findings. And then again, to show the importance of doing GWAS and then in the context of disease as a host pathogen interaction is that doing these GWAS actually help us to do things. GWAS for 500 patients, you find over hundreds of new HLA. So that shows that there's so much to explore, in the African genome. But most importantly is the fact that we're able to establish the reason why some people respond and some other people will survive and other people will not survive this virus. And then we found that alpha-dystroglycan, which is, a protective receptor for all world iron virus, definitely we'll have to work with a large protein. So we were able to find one thing here is the fact that, alpha-dystroglycan and large, all come together often to ensure that, facilitate the entry of the virus in the cell. But be patient who were not sick when they have the virus is because they had a mutation at large, and then when they had a mutation at large, that actually altered the alpha-dystroglycan large, complex, and then that will actually block the virus from coming in. And this based on functional analysis. But then in addition to that, then you also have to face one other question is, what happened when you got several kind of people who present the same symptoms, some are negative by your PCR, and then you realize that, they're presenting symptoms similar. Remember we were saying, one of the common denominators is fever. So we basically also went on to create the first largest metagenomic surveillance study in West Africa, and basically categorizing people with febrile illness in different cohorts. So people in hospitals, in communities, and then those people, and then also individuals, that do present with symptoms like Lassa fever. And then use them for metagenomics, and then that will help us to, along the line, help us to make some breakthroughs. So we discovered 2 novel viruses, for instance, like we say Equimovirus 1, and we see AKV1 and AKV2. But then what is interesting about that is, the interesting thing, as you see here, we're looking at that in quote, healthy individuals. We were doing a case control study where we were saying the fever was actually going to be the ones sick, and then the ones without fever will be our healthy people. And then we realized we found these 2 new viruses in people that were healthy in quote. And that was very puzzling and strange. So we went back to the communities. Not only did ELISA in the communities where we found this individual, 87% of them actually had exposure to the virus. But these 2 patients that actually had this virus, we were wondering what really was the problem. How come they didn't have a fever? They were in the healthy group, in quote. And we realized that in that case, they admitted they were taking what we call natural or traditional concoctions, that has high level of antipyretics. And that's the reason why at the time when we were sampling them, they didn't have fever. But yet, of course, they were infected. But then fast forward, this is a case where, again, to show how this technology could be very powerful, in November 2018, we got, 179 school children dying in the southern part of Nigeria of a mysterious disease in quote. The Nigeria Center for Disease Control took into, did all they could. Look at malaria was negative, Lassa fever was negative, all of the other things that were negative. And then they sent 22 samples to our lab, and then we went on to do metagenomics. And within 72 hours, we found that it was a yellow fever. But then this yellow fever strain was different from the one that had been circulating. You could see them closer here. And these were the ones that was previously circulating in Nigeria for the past 94 years. This information was given straight away to the Nigeria Center for Disease Control. They declared an outbreak, and then within 2 weeks, they were able to contain that outbreak locally by isolating and then creating some very local very basic ways of doing this, and then in that process, saving thousands of lives that could have gone. But then this is another case where, for instance, we are talking about a disease that is very prominent in the U.S. and then it was thought not to exist in Africa. This is a case of Candida auris. Well, we thought we were very localized here, but we had a case of a situation in Lagos, Nigeria, where we had 2 patients dying in little or no time, and then the samples are sent to our lab. And within, we went on and then established that it was it was Candida auris, and it was the very first case, described in West Africa. Again, that communicated with national public health institutions, and then they were able to contain that. But then the same apply for, extremely extensively drug-resistant tuberculosis, where we described for the first time, in Nigeria and then in West Africa. So, but then, the thing here is when you have all of this, often we are confronted to many problems in the context of Africa. You're thinking about having access to all the databases, and having access to computational databases and bioinformatics, but then you got a question. So if you don't have access to internet, if you don't have access to the database, what do you do? So I assigned 2 of our students to actually create, analytical pipeline to assemble genomes, and then also to do that for both, viruses and bacteria. So I think Paul up here, I was told to create a platform or an analytical pipeline for viruses, and then here, was to create the one for bacteria. So we successfully established that, did that, and then that provided a unique opportunity for students in Africa to download these apps and then have access to them, and then do the work on their laptop without really having to depend on these huge databases, even without internet. So that, again, putting it in context, creating tools that could be very helpful. So what did all of these things help us do? So I think this was, again, we talk about stories of 2019. So what did it help us do is actually help us to prepare what I call a pandemic preemption and response system. So in 2018, through the Audacious project, so there was a call for grant, and then we apply. And then I think in 2019, toward the tail end, we were told we were successful. And I do remember I was in New York in early January, end of January, giving a TED talk, and then as part of this program, and then I hear the very first case of COVID-19 actually striking in the U.S. And I told my colleague, you know what, if it's happening here, I need to go back home because I know we're going to be in trouble very soon. So I left the TED talk and then went back. And as we rightly said, so we were And then we got ready and then prepared for this. And again, this was the second time. For me, it's always like anytime we want to set up something, we get tested. We got this program established, got funding. We were barely a month after our funding process. And then we were told, we got to, you need to show us what you can do. So we went back and then got prepared. And we started having the first case of COVID in Egypt, in Nigeria, a few weeks after I came back in Africa. Then the second country that reported COVID was South Africa. And Nigeria was a third country. But then the difference between Egypt, South Africa, and Nigeria is that while Egypt is still struggling and trying to understand what to do with it, South Africa was having challenges to understand exactly how to manage the situation. Within 48 hours in the case of Nigeria, we actually sequenced the whole virus, and then we knew the origin of the virus, we knew where it came from, we knew that it was on community transmission, we provided information clearly to the government, and we knew that that was the important case. And then, importantly, we shared this protocol to all African countries. And then this is a protocol that you see African countries going ahead to use to discover the beta variant, to discover the Omicron variant. It was basically based on what we did, and then also we went on to train, people as we do it. We're building a flight while flying. But then this really, because the fact that we did this in such an incredible speed, actually built confidence and boldness in all the colleagues to realize that they could do it. And that really actually put the continent in a place whereby we could actually work and share the data with the rest of the world. So we're the second group. I think there was a group here in the U.S., and that is the U.S. CDC, to describe this very first variant. And then we didn't know, as we were putting this in virological, we realized that they put up their own post the day before. So we're the second group to identify this variant and then share. But what is important here is the fact that to show here in this slide how you could use data to help. So this is Nigeria, and then you could see in real time how we were detecting all these variants and sharing the data with the government, what we call the Presidential Steering Committee, headed by the Vice President. So, and I can tell you that, at different time point, we knew which variant was circulating in Nigeria. At some point, we know we got 49 different variants. And there was no way the Presidential Steering Committee could hold a meeting without data from us. So the decisions in country were actually guided by a lot of scientific evidence. And then you can see here over time as we're characterizing this. But most importantly is the fact that we went on, leverage what we knew before, and then actually use the data actually to create a CRISPR diagnosis for SARS-CoV-2. You can see here the very first. It's very raw, right, in our lab, just to show you exactly what it looks like. But we've gone ahead to work that, and we'll have that prototype here now, and then it's power-free. And then you just have to put saliva, and then you can get that in. But again, we go on to challenge with the chief immunologist in my group to ask again, Chinedu Egu, and then tell me exactly what is going on. And he went on to actually show us clearly that, yes, we had a SARS-CoV-2 in the country, but then what is important about what he found is the fact that, we look at people that were exposed, admitted, and then those that were sick, and then those that were in community. And then the level of T-cell response was very high in those two groups, so which means that there was a lot of exposure, to the virus. And that really was very helpful. But then what do we do with all this information? So it's all about training. So we shared this, train people in different African countries on how to do what we're saying. And then over the years, within that same period, we've trained, as Charles mentioned, over 2,000 young African scientists in 53 of the 54 African countries. And that, you could see, that has a huge impact on how this continent was actually using genomic data to respond to the epidemic. I remember at the end of 2020, John Kengerson, who was the director of the Africa CDC, called me and a few people and then to say, what? We're not doing enough. We need to accelerate the way we're doing it. I say, yes, sir. But, the reality is the fact that at the end of 2020, we got barely 5,000 sequences on the continent. But then because we train and then we're able to provide, share information, and then share the skill and knowledge and technologies and the protocol, by the mid of 2020, by September, we've had over 60,000 sequences, and then at the end, we got more than 100,000 sequences. And those really became very important, because not only we were sequencing to help public health response, but we're also making discoveries, as you know, beta variant, Omicron variant, helping diagnosis, helping, vaccine development. But we also learned some great things in the midst of that outbreaks, that if you're looking at responding to disease outbreak, remember I've been spending all the time talking about what happened in human. We learned quite a lot, that if you're looking at only human, then you're really looking at a very small part of the problem. So we got to be very more holistic about the way we're looking at it. So, and then we recruited, Professor Anne is happy here. So we actually brought a lot of dimension and innovation in the way we're doing things. And one thing she did during that period is actually not just to look at So start really using one-head genomic surveillance to characterize pathogen only in human, animal, and in the environment. And that really became very helpful because looking at the environment, looking at human, we discovered a few new things. Looking at Lassa fever alone, we discovered new hosts for the virus. We thought initially that the mastomid natalensis were the only hosts, but we realized that there are many more natural hosts. So which means as we are looking at problems, if you are just very narrow, at the end of the day, actually the source of infection might be just next to your leg, but then you'll be looking elsewhere. So that really became important. We're looking to bats, trying to look at what is going on in a part of the surveillance. And, but we also learned a lot during COVID, as you all know. We realized in the case of Nigeria, so when we have a flare-up in human population, we also observe a flare-up in animal population. And then when the number of cases dropped for human, then we also saw that. So in real case, there was a case of reverse zoonosis, which means that we spend our time infecting our pets and infecting our animals. And then when we have that kind of situation, then they become a bit scary and then worrisome. So, I mean, and in this case, for instance, we say that yes, while we're doing this, then you really want to start sampling bats and other things, and we discover some new, alpha and beta coronaviruses circulating in bats. But let me move forward to the end of this epidemic in 2022. Then we were dealing with something else, which was Mpox. We all know that the global Mpox, became a big problem, a big issue, in the sense that, we had population infected across the world. But then the reality is that, Mpox has been circulating in Africa, or have been creating havoc in Africa for years, but nobody, or what I call the regional health organization never bothered actually to address the problem. But then when Europe actually got affected, then, there was a huge mobilization of vaccine, stockpiles of vaccines and everything. And then that really led some of us to go into the global database and use African, genome sequence data to actually make some meaningful analysis. And then we came up with this. And this is what? Showing clearly, as you see here, that this was a brand new lineage, right? And they're responsible for Mpox transmission and spread. And as we know, we are talking about taxonomy of viruses. Then the question was, okay, so we are all talking about all the time about the West African clade, or the Central African clade of the virus. And then my question to the WHO at the time was, okay, now that we all know very much about the Central African clade and the West African clade, and then we'll have this new clade. How do we call it? We call it the European clade. And they said, oh, no, it's not possible. I said, then we got And then I said, well, if you cannot call it, European clade, then we have to come up with a nomenclature that is more neutral, that is less discriminatory to everybody. So I think eventually, after a lot of argument, so they were able to agree, because I said, if you don't agree, then the data will speak for themselves. So I have a name. Then if you don't want to agree with this name, then we'll cancel all names. So at least WHO, of course, had to go ahead and cancel all names. We'll talk about what clade 1, clade 2, clade 3, or whatever we call it now. But actually, they were not. They didn't do it because they wanted it. They were forced to do it because we have the data. Again, to show the power of data here, in order to actually to be, we're talking about equity. We're talking about, for equity. So fast forward, so we really spent the time to study this virus and then use this as a way to do this in West and Central Africa. We brought all these countries that reported Mpox together and then studied this to really understand what was transmission dynamic, phylogeographies, and everything across the continent. Importantly, we actually showed that, this B1, clade that I mentioned here was from Europe and was following transmission in some African countries. But most importantly is the fact that we established the fact that that flare-up that we saw in 2017 in Africa, a wolf from Nigeria probably spread elsewhere, actually started in 2012. Cryptic transmission going on without people knowing, as you see here, all the way from 2012, and then by 2015, we tend to see, it peak. But then we also identify that it's not going that much, because you could look at the exponential growth. It's really not that fast. And that's the reason why the spread wasn't that bad. But then the doubling time was about 2 years. But importantly is the fact that we established that it was followed by human-to-human transmission. And then in the case of zoonotic transmission, we look at the APOBEC, and we realize that the APOBEC was 40 times. So that is basically having 40 mutation much more faster than what it's supposed to be. So in the case of Nigeria here, you could see transmission over time. We're establishing transmission chains. We see over the years that the transmission was going on no matter what happens. And that really was very helpful to help the government. We established hotspot, and then the new Port Harcourt were one of the hotspots from Port Harcourt spreading across the rest of Nigeria. And that also was useful for government to actually start using to address the problems locally. But along the line, we discovered, like a new clade, which is a clade 2B, and that is from zoonotic transmission from Nigeria. But then that clade actually is not just circulating within Nigeria, but also has to do with circulating across the border with Nigeria and Cameroon, as we see here. And, we went on to work with the Cameroon team, and we were able to establish the origin around southwest Nigeria, but also have that transmitted across border. So which means in real sense, instead of, we need to be very holistic and look at cross borders, studies, instead of doing the kind of work that we do in silos, but let's do cross-border studies. But most important thing is that we use all of this data to address one major problem. One thing that you observe in the field is the fact that, the time that it takes when you are doing the kind of work that we do, getting the data in the field and then sending that to the ministry, it takes such a long time. And then even when the ministries have the data, and then even the national public health laboratories, they don't have the power to actually do the kind of work that we do. So we've always been thinking about creating a system that can enable national public health institutions, laboratories that do not have all the kind of computational power that we have, to do this analysis in a very short time. So through the Sentinel program that we have, we got one of our postdocs to actually go on and create something that is revolutionary. And then Patrick went on to create what are called Delphi. So you're going to see that popping out in Nature very soon. So creating this, is set up an application for those that are in field of bioinformatics, you see, to do Bayesian analysis. If you look at what Bayesian, if you want to go to a BEAST analysis, it takes such a long time. The point is, you want to do this thing within weeks. That's what will happen within weeks. But in this case, for instance, we're basically talking about plugging in data, and within 5 minutes, you can have the result. So imagine taking all of the data that you have in GISET, hundreds of millions of genome sequences, and then get this thing done to you in 45 minutes. That's the kind of feat that we've achieved, through this program. And this is now open access, is now being shared. But again, just here to show that, we created a tool that can help public health institutions to respond in near real time. In addition to that, we created a dashboard, that can do GPS data and then all information that can be provided. So, but when you're dealing with all of the diseases that we created, for instance, so what do? You really need to think about creating a platform that can help you to do all of those analysis. So we've gone out to create what I call CAMEN, which is a combination array of multiple CAMEN tests. And we created panels that can enable you to do up to 49 different pathogens in one single test. And then all using CRISPR. So, and then that really is really going to be a game changer. You can see this in my lab right now, because we believe that with this, you should be able to actually test and validate, one single blood sample, to look at all these different pathogens, be it parasites, bacteria, and then, and viruses. So that really has a lot of power. But at the end of the day, what we're trying to tell here is a story whereby we believe that you could have all the genome center, but most importantly is actually to just use this genome data to create tools and technologies that can be moved down to the field, work with the communities to refine those tools, and eventually, establish a sustainable and independent, surveillance outbreak response system. But then, how do you do that? You really have to get the infrastructure. You need to have the human resources. You need to have what it takes you to do that. So we're basically setting up a model, and that model is what we try to experience here. So Charles was mentioning today about this Genome Institute. So this is a genome center we just created. You can see there Charles and others. You can see him here presenting with Charles. But then also Chinedu here receiving his award for best presentation. I want to believe that you guys are doing a fantastic job at NIH. He has got to come over and then get the best overall presentation. Come on. So next time, I think we're going to prepare the kids much more better to be better than you. So what we're trying to use this as a model. Have a genome center like this one, but then that becomes like a hub. And then we set up a hub and spoke model, where this hub has a mandate to mentor, to build other developing centers. So at the end of the day, we can have a lot of hubs across the continent. So basically, what we're trying to set up is what I call, the Network of Genomic Centers of Excellence in Africa. So it's actually pioneered by myself, Nicola Mulder, and Ambrose Honkamp, and John Hopkins. And it is basically just, built on two models, hub and spoke. And that's why I'm talking about lift as we rise. That is, have a hub that has a mandate to lift orders, and then build these centers of excellence. And then at the end of the day, we'll have 10 centers of excellence that will have a mandate to build another 20. So if in Africa we've got 30 centers of excellence, I think we should be able to cover what is needed. So, and that really gives you an idea of what this whole thing is all about. That is basically looking at very important issues, pathogen genomic surveillance, genomics to inform precision health for public health, and then workforce development. So we believe in that, through this learning from COVID, no matter what you do, there's actually one major vulnerability that Africa still has. We could do all of the great genomic work that we want to do for surveillance. The reality is that we don't have access to vaccine. We don't have access to diagnostics. So if you have the next pandemic, we're going to be in the same situation. We need to create a platform that will go beyond just doing this by being translational into the way we're doing it, going from genome sequences to diagnosis, going from genome sequences to vaccines and then to chemotherapies. And this is not just looking at infectious disease. We also want to look at non-communicable disease and others. So I think that basically we're creating things that is coordinated in, a network of centers of excellence, coordinated and driven by African scientists for equitable capacity workforce development, but also to work with partners, to bring in, advanced genomic medicine and work with the industry to support the future of translational genomics in Africa to actually create, through an equitable mechanism, which means that we want to work together with partners who are sharing the same vision, and then create the next generation tools that is actually needed, not only for Africa, but for global health security. Because we believe that through the biodiversity in Africa, we can actually, make a big difference. But how do we want to do this? This is a value proposition. So we want to create an ecosystem. We know that clearly in Africa, there's just many, many initiatives, but they're all fragmented. Can we work toward bringing all those initiatives together in order to actually work in pandemic preparedness, to workforce development, research discovery, and then others. But then the value proposition here is that there've been no initiative like this one in the past. Everything is all about doing different things in silo, but in terms of translational research, they've really never been one before. So we really want to go beyond where we are now. Let's go a little bit beyond. Let's be translational. Let's use the information and the knowledge that we have for both human genomic sequence data and infectious disease research to create the next generation diagnostics and the diagnostic tool. So we've done the landscape analysis. We know exactly what is available in the continent. We know exactly where, the platforms are. Yes, we know. We've also gone ahead to actually identify, in the geographical landscape, what are the different initiatives that are going on. And then we really want to bring them together. But then also we're talking to, a lot of potential investors, as you see here. So we're talking to the SCORE, Wellcome Trust, and others, MasterCard, and they all have different interests. Then the whole idea is to bring everybody together and then see how we can, bring your different interests together into one particular umbrella and then see how we can use that in order to do one thing and to do a few things. And those things are really actually to change, the way the health research and then the outcome of health research in Africa. And then some of those things are what? We want to create a long-term workforce development. The reason why we're talking about a lot about long-term workforce development, and then I'm happy the Fogarty Training Center is here. They played a critical role in developing the career of some of us. But then we need to remember that right now, Africa is 1.4 billion people. In the next 20 years, that continent is going to be 2.5 billion people. And it's going to be the largest and the most populated continent in the world. We are talking about precision medicine and everything. So that is going to be the epicenter. And for health development to grow, we need to start sowing and building the workforce in the country because it's going to be a center of gravity. It will shift, and we need to start preparing that workforce right now. If we don't do that now, we'll face a major challenge in the future. We'll have all of the technology, but we won't have the people to work. So I think this is a time to invest seriously into that. We need to work on local manufacturing, local diagnostics and vaccine development. We have all of the platform. We heard about Moderna, Pfizer, putting the platform across Africa. The question is, where are the people to work in? So we need to build to work for. We need to train the people. Otherwise, those platforms are not going to be useful. And, Charles, I always take this example of April 1. From Charles' work, very incredible work to say we don't have that many African genome represented in the global databases. But this actually is an example of how you could use genome data to address a problem with kidney disease, for instance. We believe that we can actually find a cure, through this kind of program and realizing an AIDS-free generation. Deliver a cure for sickle cell. We all understand clearly that, yes, there is, but we need to make it cheaper. We need to make it more affordable. Can we do that? The answer is yes. So how can we do that? By creating the ecosystem that we're talking about, by actually just nimbling the technologies and then and do things much more faster and cheaper. So understanding the genetic risk of cardiovascular disease and then use that as a way to create the next generation solution. Establishing long-term clinical trials. Of course, we're having all of these new solutions that are being created. The question is, where are we going to be doing those? We need to establish the clinical trial centers in order to do this and then move these things to the field. And eventually, precision oncology for Africa is actually going to be a big deal. So really, I think it is, I do hope that I'm making the case, for this new initiative. But then I really want to really thank this group of young folks that are doing the work and extremely well, and also acknowledge that are actually supporting our initiatives. So thank you very much.
Please, anyone that has questions, use the mics. There's a question online, but that tour de force at the end, I think, more or less answered a couple of these questions. Unless you want to augment it in a little bit. What are some intentional, bi-directional strategies to promote continuous learning training programs, research collaboration, data sharing, to optimize global genomic sequencing techniques? That came in midway through the talk, but again, at the end, yeah.
I do think that, it is very much achievable. But in order to do that, we need to invest in the right places. So, I mean, this investment has to be done not only by global partners in the North. The African countries have to make the investment themselves, and then we need to have strong partnership. So I think with that, we should be able to actually create what I call what is needed. And then what is absolutely needed is actually also creating the environment and then the necessary incentive that people need in order to build this capacity. There's no point, saying that we want to do this without having what I call creating the necessary incentive. As long as we have young Africans that cannot put bread on the table, it's going to be very difficult. We have to be very intentional about making sure that, people that the salaries and things are competitive. We also have to make sure that we're creating environments that are, working environment that are competitive enough. For instance, for those that have come to the institute where we are, we really did not think twice. We put in all our best and we made sure that we created an environment that is very competitive in terms of infrastructure, very competitive in terms of anything that you can imagine here. So I think a student or a postdoc working in this, in our own environment, coming to NIH is not going to be impressed, right? He will know, there's no big difference. So I think we need to actually be intentional about creating, the necessary incentive that is actually needed for that.
Mm-hmm. Yes, we have Jennifer.
I can't resist asking a question. Beautiful work as always, Christian. And I think sometimes as scientists we feel a little powerless politically, and I think that you've really brought to the fore the need to engage World Health, Africa CDC, local governments, other scientists. So what is your message to young people getting into public health on how you do that effectively, because you have been very effective?
I do think that, one major challenge that we do have as scientists is actually often to connect with, as Jennifer mentioned, with policymakers and then people making decisions. But in order to connect very well with them, you need to have what I call clear-cut use cases. You need to have a use case that actually show them that there could be, the investment that they're making, for instance, has a direct impact. And it's not about doing the science for publication. It's not about creating a lot of papers. It's about showing that the work that you're doing save the lives, and then doing the work that the work that you're doing matter most for them. I give you instances for during COVID, from 2020 to 2023, we did all of the work that we did. We had no single publication. We identified all these variants. All these data were given to for policymakers and politicians to respond. They could see that. Our paper for COVID actually was almost rejected because it came out in 2023. So, and I think they could see the value in the data that you're providing. I'll give you instances, for instance, in the case of Nigeria, you say you were in a lockdown. We had a lockdown. But of course, because we're giving reports on week to week, you realize one variant that was in Lagos, and then the next week you are seeing that variant in 400 kilometers away. And we were like, what is going on here? If this variant was here and then there's a lockdown, there's no reason why this variant should find itself somewhere down there. Of course, we said it is very possible that people are moving. And we told this to government. Of course, what they did is that there was a task force. They set up another task force to oversee that task force. And, and guess what? They realized that between the hours of 11 p.m. in the night and 4 a.m. in the morning, people were on the road. That was very helpful for them. They could see the value. They don't understand the whole thing about genomics, but at least they could see the value of the data. And that, for them, is very important. So as we're doing the science, let's do things that people can see and then realize the impact. And then, subsequently, if you tell them to invest, they will not say no in investment.
Thank you. Please introduce yourself.
Hello, my name is Oyebola Ishola. I was part of the trainings in NIAID, in SGID at some point. I actually did an internship. But now I'm at NIAID. My question is actually so you kind of described this to a large extent, looking at the virus. How much work has been done in looking at the host side, like the immunology side, and how this might be influencing some of the differences we're seeing? So you kind of mentioned some of Chinedu's work, but like sequencing-wise, looking at innate cells, how much capacity do we have to be able to do this? And you've talked about talking to partners and kind of pitching in. To what extent has conversation gone into building this part into the new institute as it's getting bigger?
Yeah, thank you. I think we are getting there gradually. One of the things that we've done strategically is to, in the immunology part of it, I think we want to go beyond just understanding what neutralization and the T cells things. I think Chinedu is here learning how to create monoclonals and everything. That will have a huge impact in terms of, chemotherapeutics and then potentially vaccines. So I think we really, but then that comes, with also understanding the repertoires of immunology cells, and then we basically just made also very huge investment into single 10x genomic single cell sequencing. And that will help us to really go into, the nitty-gritty of understanding exactly from the immune side, immunology side, or what to do. And I do believe that, when it gets back, so that will be very complementary to what we're doing, and that will help advance the field in a big deal. So I think, in another dimension, I think I need to start asking for NIH to pay us some money back, because we train all these young people and they end up here. Come on, so she's here and then Chinedu is here. I think we need to think about what is the sharing formula, right? Thank you.
Thank you very much.
You can tell that he's not very shy about that.
Please wrap
You
up.
have questions from
No, please.
Okay.
Wrap up.
Again, in my excitement to introduce Christian and Anis, I forgot to actually tell people who I am. I'm Charles Rotimi. I'm the scientific director for NHGRI, and it is indeed, as I said in the beginning, you can see the kind of fantastic work that is going on there as having impact on people's life. It's creating new science, new technology, but more importantly, it's really saving lives. I think that's really sort of the bottom line in everything that we do. So I want to thank Christian and Anis for coming here to share with us the wonderful stories. And I promise you, you're going to hear more in the coming days and coming months and years. So thank you, Christian, and thanks everybody for coming.